Emily, you mentioned before Cartesian's unique mRNA approach to CAR-T manufacturing. And so vector manufacturing is a big part of CAR-T manufacturing. And at least for the foreseeable future, we will need vectors to manufacture CAR-T cells. So I'm curious, from Cartesian's point of view, how does the mRNA approach to manufacturing not only cut down on, why mRNA approach to CAR-T, and how does this differ from traditional integrating vectors? Let's start there.
Sure, happy to cover that a little bit. And I know we're going to talk about viral vector bottlenecks, and I would say that the best way to eliminate a viral vector bottleneck is to not use a viral vector. And so that's the approach that we take. There are actually other reasons why we do this. As I mentioned, we do mRNA cell therapy, mRNA CAR-T specifically. mRNA is a transient modification to cells. So as cells that are engineered with mRNA are infused into a patient, they begin to divide. With every cycle of division, the mRNA is diluted out, and then eventually it goes away altogether, and then the resultant protein expression also has a decay curve associated with it. And so our thesis is that you can dramatically improve the safety and potentially really expand the indication space that you can address with a CAR-T cell if you take this mRNA engineering approach. And so we've talked a lot about scale. Typically the lentiviral or viral vector transduced CAR-Ts are dosed at a few 100 million cells. We dose a couple of orders of magnitude above that. And we're aiming to deliver a therapeutically effective dose to the patient at the time of infusion that then doesn't rely on those cells proliferating after they're infused in order to achieve efficacy. And so we deliver without lymphodepleting chemotherapy. We deliver in an outpatient setting. And as I mentioned, we've just actually reported top-line results of a phase 2b placebo-controlled trial. Turns out you can execute a placebo-controlled trial with an mRNA-based approach, which has its own challenges when you're talking about a viral vector modality with lymphodepleting chemotherapy. So lots of reasons why we're really excited about this mRNA-based approach, and think that, to Jason's point about it being it being a new modality, it is, and we think it actually is perhaps a pretty elegant solution to bottlenecks and access to viral vector. So I'll stop there.
Including supply chain, right? Right. It should be the supply chain for an mRNA is much easier than.
Absolutely, and very scalable manufacturing process for mRNA. So pretty efficient. So a lot of advantages.
Yeah, one of the biggest challenges with vectors. I mean, not scientific challenge, but just I think it has gotten better. I think you can wait for like 6 months now for a new lot. If we talked a year or 2 years ago, it could have been 2 years. So definitely, I think that's a big challenge with viral vectors. It's just getting them quicker enough, and the whole supply chain challenge.
Yeah, Tal, would you say that vector, to what degree does vector manufacturing impact overall CAR-T manufacturing time?
I mean, it shouldn't, because it should be one of those things that are off the shelf, right? You should, if you have a good enough supply chain, you should think about these things and have enough vials sit in your freezer, given that the stability is acceptable, right? But during COVID times, that was a big challenge. Just getting enough and making enough for one lot. How many lots do I need? Sometimes you can only make a few, maybe make a dozen products from one vector lot. So that's another area of much needed innovation. Just getting the titer up and scale up.
Yeah. Great. Thank you.
With Lenti in general, it just extends your supply chain, right? Because you need, Lenti is almost exclusively made transiently. And so then plasmid DNA is part of your ongoing supply chain. And so it just makes your supply chain like lots of elements of it. And the longer your supply chain is, the more chance there are for hitting bottlenecks at any time during manufacturing.
Yeah.
Great.
So make
Thank
all
you.
your vector changes early on and lock the process. so you can
Good advice.
use all those things you have in the freezer. You don't have to throw them
Easier
away.
said than done, Tal.
Everyone write that down in the audience. Do it early. Simple. Well, great, guys.